My name is Michael Raiche. I'm that senior portfolio manager that Lindsay mentioned. And I'm joined by Jerry Zion, who's our global training manager. And today we're going to walk through the topic, putting your finger on pulse oximetry. So we'll go through pulse oximetry at a few different levels to make sure that we understand what's going on, why we're doing it, and what the technology behind it is. So let's jump right in. So to start, what is pulse oximetry? It's the measurement of the oxygenation of arterial blood. So pulse oximetry monitors a term called SpO2, and the acronym SpO2 stands for the saturation of peripheral oxygen. That's what you're going to see on most medical devices. The amount of saturation means how much of the arterial blood is carrying oxygen. So 100% saturation would mean that all of it is oxygenated. The pulse oximeter reads out in percentage of arterial blood that is oxygenated. So you're always seeing it as a percentage. Biomedical equipment professionals should be well grounded in the fundamentals of this very valuable technology in order to understand it adequately. So we're going to start with the basics and then build up to cover all the details. Jerry, you had a thought on this in terms of hemoglobin before we jump on to the next slide.
So the specific molecule in the arterial blood that we're concerned about is, think of it as a boxcar or a truck, a lorry that is going to carry something. And in this case, it's the hemoglobin molecule, and it is supposed to carry oxygen. The problem is sometimes it gets filled with something else that is not healthy for the human body. And so we really do want to know the percentage of saturation of oxygen in the hemoglobin molecule. And we'll talk a little more in detail about that as we go through.
Thanks, Jerry. Yeah, so jumping into hemoglobin, in the red blood cells is a protein called hemoglobin that Jerry just touched on, and that's what carries oxygen to the body. So in that blood, the hemoglobin is actually oxygenated or not. In a pulse oximeter, we'll measure the percentage of hemoglobin in the arterial blood that is oxygenated. So pulse oximetry is a very important measurement for patients. It gives a very quick determination if patients are having trouble getting oxygen into their bloodstream. So if they have trouble, it can be treated as soon as possible. Now, what we're focusing on here is pulse oximetry, but there is also a technology called pulse co-oximetry. And Jerry, do you want to put some color on that?
Yeah, so as I said, sometimes these hemoglobin molecules get filled with something else, and that something else is typically carbon monoxide. That's a poison to the body. So unfortunately, pulse oximetry is using just 2 wavelengths of light, and we'll get into the details about that a little bit later. Whereas the pulsed co-oximetry is using 8 or more wavelengths of light so that it can actually identify the difference between what is oxygen and what is carbon monoxide. And so it may or may not be a technology that has reached hospitals where you're working, but it will, because it is a non-invasive way to understand more, especially more about oxygenation of the blood than we could ever do with just pulse oximetry. So we'll go more into the details about all of this as we go through this presentation. So we just want you to know that there are 2 technologies that may be used, and the one is actually more capable than the other, but the pulse oximeter is the most likely that you're going to see right now.
And that's a great point in general, is just medical devices are becoming more and more non-invasive. So the less we have to do inside the human body, that's the direction that the industry is going. So the more we can learn through light and coming in from the outside of the human body, that's where we're going to go. And that's where pulse oximetry will continue to expand in the future. So here's the kind of the first dive into the technology, because we know that arteries carry oxygenated blood throughout the entire body. We know that arterial blood comes from the lungs, where it's picked up that oxygen and it gets pumped through the left heart chambers to the aorta and then to the arteries. And that arterial blood moves in pulses as the heart beats. So pulse oximetry, it measures the oxygen in these pulses of the arterial blood in a peripheral part of the body, typically a finger. So the pulse oximeter can work quickly, so it gets a result in just a few heartbeats. It checks that the arterial blood is indeed carrying oxygen and will spit out that percentage. So pulse oximetry can be measured with multiple types of devices that can attach to a finger, as in the example I gave, but also a forehead, a foot, or even other areas. And we'll jump into that a little bit more.
Okay, keep going.
So when we look, we were talking about, so here's a picture of a foot, and where it could be hooked up to an infant, since children are notoriously difficult to put something on their finger and keep it on their finger. So the kind of the mechanical interface changes based on who the patient is and what you're trying to measure.
By the
Other
way,
ones that are more common are on the forehead. If you
yeah,
think about a technology we'll get into later called reflective SpO2, where that technology is starting to rise as well. Go ahead, Jerry.
interestingly enough, pulse oximetry, one of the first places that it was typically measured in the clinical situation in the hospital was using an ear oximeter. So this oximeter was like an earring. It clipped to the earlobe at the bottom of the earlobe because there's pulsatile arterial blood flow there. And it was used by respiratory therapy or to assess and give an estimate of perfusion, just an estimate. But that was the first place. So you may see a little clip thing, a pinch clip that would clip onto the ear. That was for an ear oximeter, a measurement at the ear. So fingers, toes, heads, and earlobes, okay? If you're thinking about the where do we make these measurements.
All right, so I'd like to back up just a little bit and jump into the metabolic cycle. I want to walk you through how the human body metabolizes oxygen, and I'm going to walk through this circular diagram here. So the first step is that oxygen is inhaled. Then that oxygen goes into the lung, and the lung oxygenates the blood. And remember, this is the hemoglobin that gets oxygenated. The blood then gets pumped and reaches the extremities. And that's where you're going to get your pulse oximeter to measure. And then deoxygenated blood then comes back to the lung to be exhaled as waste gas. So if you think about it, we're bringing oxygen out to the body and then waste gas back to the lungs to be exhaled. So there's a whole measure here. There's another medical device called a capnometer, or an EtCO2, end-tidal CO2 monitor. And those measure breath by breath. And this device measures how the body is metabolizing oxygen into CO2. It's a more instantaneous measurement of oxygen metabolism since it is breath by breath. So any changes of the patient condition will be delayed on a pulse oximeter since enough blood needs to be pumped from the lungs to the heart to the finger in order to get that change to register on the pulse oximeter. But it is breath by breath on a capnometer. So end-tidal CO2 really measures how much waste gas is leaving the body, or is oxygen being metabolized, whereas SpO2 measures how much oxygen got into the bloodstream. One of the terms here that we'll jump into is perfusion. And Jerry, if you want to add a little bit of color there, that'd be helpful.
So perfusion really talks about blood flow, and blood volume. Because with each contraction of the heart, there is a volume of blood or a cardiac output that is the volume of blood that is moving out to the periphery, to the blood vessels and arteries and to the cells. So when we're worried about supporting healthy breathing in the patient, we want to know both sides of this. The oxygen side for oxygen coming in to each cell in the body, and especially those that are at the far distant points in the body, fingers, toes, heads, and nose, those kinds of things. And we also want to know how much carbon dioxide is being exhaled. That's called gas exchange, and it is the goal of proper gas exchange is to get oxygen maximally in and carbon dioxide out. So what we don't want to see is any inhaled carbon dioxide. We want to see it all going out. And so it's a measure of the health of both the blood interface with the lungs and the lungs themselves in terms of getting this gas exchange in a proper range. So it's all part of the picture. Today, we're just focused on the perfusion side. That is the part where blood leaving the heart is full of oxygen to distribute to each of the cells in the body, and then leaving those boxcars open to collect carbon dioxide so it can be exhaled by the lungs.
Yeah, both technologies give you that full picture of what's going on with the body's metabolism, but today we're going to focus just on SpO2. I want you guys to make sure that you know that there are other technologies out there as well that do call it different things to give us a different view of the same or a similar problem.
Well, the cool thing about all of this is we're talking about non-invasive measurements that can give us a really good picture of what's going on that are not able to be identified or understood by the five senses, okay? What I can see, what I can hear, what I can smell, what I can taste, and what I can feel. You can't get that understanding of this gas exchange with the five senses. So it's technology that is really enhancing or really giving more than just that five senses assessment of patient health. And that's just so important.
So I'd like to jump into a little bit of the history here. And before pulse oximetry, there was really no quick way to analyze the oxygen content of blood. And instead of the non-invasive way we do it today, they could do it invasively by drawing real blood and then running tests on it. And that took time that patients couldn't necessarily afford, especially on the operating table under anesthesia. You have a patient system that's under stress. So before pulse oximetry, thousands of patients died of undetected hypoxemia, which is a low level of oxygen in the blood, simply because of the time it took. Now the pulse oximeter is ubiquitous and can be found in most hospital rooms and emergency facilities. This blood oxygen level is routinely measured. Doctors and nurses know immediately whether the blood oxygen is low, and it lends itself to diagnosing a patient. So this pulse oximeter, although it seems very simple and ubiquitous today, it wasn't before, and it's a great device to have available, especially when forming a patient diagnosis. Jerry added a note when we were talking through the slides that the man behind the technology is Dr. Severinghaus. He kind of innovated and brought the technology into the space. But it's good to know where we came from and that we were invasive at a time. It had its drawbacks, and we've been able to move in more of a non-invasive direction, and patients have benefited because of it.
Well, and indeed, we still do blood gas draws. We actually do get samples of blood, take them either nearby to a blood gas analyzer or co-oximeter, or go to the clinical lab wherever it is in the hospital with that. And both end tidal CO2 measurements and pulse oximetry measurements are compared with the gold standard of blood gas, and that's part of how Dr. Severinghaus made sure that that we had an effective tool in pulse oximetry. Now there are drawbacks, and we'll get to some of the drawbacks too, things that can fool the technology. So we'll talk about that in a little bit.
All right, then I think this is a good time for the first poll question. So I'll read through it and we'll bring it up on the screen. But what wavelength range contains both red and infrared light used in pulse oximetry?
So this is kind of a tease. We know we didn't tell you this so far in the presentation, but we want to see how many of you engineers were really paying attention in class. So answer it if you can, guess if you need to. It's all okay. Don't worry about being right or wrong. Just do your best.
So in one of these wavelength ranges, both red and infrared light will be in there.
So we do have the poll open up on your screens right now, and we'll give it about another 30 to 45 seconds here, and then we'll read out what the answers are. And don't worry if you're wrong, it won't follow you anywhere, I promise.
No, this is just to see where people are before we dive into it. And if it's a total guess, that's fine too.
Okay, about 5 more seconds here, and then we'll close the poll and see where everyone ended up. Okay, so we have closed the poll, and we had 10% of the answerers said the first answer there, A. 27% said B. 56% and the overwhelming majority said C. We had 4% with D and 3% with E.
Awesome. Well, I'll let you guys know that the overwhelming majority was right. So if you look in the bottom right there, red has a wavelength of 660 nanometers, and infrared is between 900 and 940. So great job to everyone who guessed that. So thanks for that. But in general, right, the light that we see isn't all the light that exists, rather just a portion. So if you think about the visible light spectrum, it's just a portion of actually all the light that's out there. If you remember high school science class, Roy G. Biv was one of the acronyms used for the visible light spectrum. So red, orange, yellow, green, blue, indigo, and violet. And pulse oximetry typically uses red light and also infrared light, which is out of the visible light spectrum. So here's how the most common oximeter configuration works. The oximeter shines 2 different wavelengths of light through the patient's finger using light emitting diodes, better known as LEDs, and they do that one at a time. So the 2 wavelengths are red and infrared, 660 nanometers and 900, 940 nanometers. Pulse oximeters, they shine that light through the human body, typically through the finger, as is shown in this diagram. But as Jerry said, it's also feet, earlobes, and foreheads. And so some oximeters reflect light off the body rather than shine light through it. And we'll get more to that later. That's reflective SpO2. And what happens is a detector on the other side of the finger measures how much light has passed through the finger from each type of light. The oximeter only needs one detector that can detect either light because it knows which light is shining at any time. So really, it's what's shown in that image there. We send a signal through one side of the finger, and we receive a signal through the other, and that and the math behind it, and we'll jump into that, tells you what the oxygenation level of that blood is for that patient.
But there's one more important requirement in pulse oximetry. The blood has to be pulsatile. There has to be a pulse that is able to be sensed. If there's no pulse, the assumption is it's just from a vein and not from an artery. So it means an improper positioning typically of the sensor. But it's not just the reflectance or the absorption. We're showing here the absorption technology. It's not just the reflectance or absorption of the light, but also the pulsatile nature of the blood that we're shining the light through, okay, the tissue in the blood. So you have to have all of those things in order for the measurement of the percentage of oxygen saturation in that blood to be valid. Otherwise, it's an invalid measurement.
All right, so we talked about absorption, right? So here's how light is absorbed by the hemoglobin at the two different wavelengths shown here in this graph. So oxygenated hemoglobin absorbs more infrared light and allows more red light to pass through. And deoxygenated hemoglobin is the opposite. So it allows more infrared light to pass through and absorbs more red light. So what's not absorbed shines through to the detector and gets measured by the SpO2 monitor. The SpO2 monitor measures the amounts of red and infrared light that do pass through, and those get detected. It calculates the ratio of red light to infrared light, and that ratio actually tells it the percentage of saturation given the relationship of each of those wavelengths and oxygenated or deoxygenated hemoglobin. There's a little more to this equation. Just like, you know, there's more to a finger than just hemoglobin that the light has to pass through. There's the rest of the finger. And this includes venous blood. So, you know, you have your deoxygenated blood, your non-hemoglobin, your tissue, your bone, your skin, and other things. SpO2 monitors are interested only in the transmission ratio caused by the hemoglobin, not by everything else. So they analyze the light transmission and break it into two components, AC and DC. So AC is the varying level caused by the arterial pulse, right, which is what we want. And DC is the average level caused by everything in total, which includes the arterial blood, but also includes everything else. So the DC level is the average measurement over the whole time, and the AC level is the change from the steady state level between pulses to the level during a pulse. So it's really just the change and focus just on the arterial component. These AC and DC components get combined into the final overall calculation as shown here on the screen. We don't have to jump into it more than that, but by calculating the ratio in this way, variable factors in the finger are eliminated, such as skin color, finger thickness, and the amount of blood pumped. Because again, we want to focus in on that AC component. We want to focus in on the arterial blood.
These are, however, some of the things that get in the way of a proper reading. So skin color, that is actually a thing. And the darker the pigmentation or the color of the skin, that makes it harder for the light to be transmitted through. Also finger thickness. So if you have a patient with a really thick finger with lots of tissue to go through, including the bone from the finger, then it also kind of gets in the way of a really good reading. And when we say the amount of blood pumped, when we get to patients with really low perfusion, this can be patients undergoing some parts of surgery. It can be patients that have injury, real serious injuries and so forth. Low perfusion also gives pulse oximetry a difficult time getting a valid and complete measurement. And so these are some of the things that the doctor or the nurse who's trying to use the measurements from the pulse oximeter, because it's non-invasive, it gets in their way of being able to do that. So what do they do clinically? They'll draw a blood gas sample, send it to a clinical lab or a stat lab nearby, wherever the patient's being treated, and get the blood oxygen level and other values from that blood gas draw. That's clinically what will happen when we have difficulty with the non-invasive technology giving us a measurement that's believable based on the other things that are happening at the moment in that clinical situation. So just understand these are some of the things that can get in the way.
So there's also another ratio, but essentially it's the same one. And this is what SpO2 monitors use. And it's basically a lookup table of ratios across a range, right, which gives you this graph here. And that's called an R curve. So R curves have been determined by clinical trials by each of the manufacturers. And since there's no scientific reference available to characterize the human body, the manufacturers have experimentally measured the ratios that will be obtained at known saturation levels. So each SpO2 monitor will have its own curve, its own R curve. They put these values into the R curve lookup table here. And an R curve will associate each point of saturation with the light ratio that it will produce. So the SpO2 monitor calculates the light ratio and then looks up the saturation level that corresponds to it in the R curve. And one thing I think we should note on here, Jerry, is accuracy.
Yeah, so the accuracy of a pulse oximeter measurement, the percent of O2 saturating hemoglobin in the blood, is based on this R curve. It's not based on any other adjustment or anything electronically, maybe some display capability on the pulse oximeter, but that's all. So it's all about this R curve, which has no metrological standards. So there's nothing to say there's nothing else to be able to use when you determine accuracy. What does that mean? That means when you're looking at pulse oximeters and trying to decide which one is the best one to use, or when you're trying to make the measurement of pulse oximetry to determine if it's functionally working properly or not, your test instrument must have a reliable R curve, which is brand and model specific. So each manufacturer of pulse oximeter, in every model of pulse oximeter that they make, they have to determine this R curve. And as Mike has said, it is based on clinical studies, in particular, because you can see this curve goes from high to low. It is really a desaturation curve. And the clinical study part of it is a breathe down study where we draw blood gases while a patient is being deprived of oxygen that they're breathing purposely. And these are all volunteers. So this is on the manufacturer of the pulse oximeter to do these breathe down studies, and that is the basis and foundation for their design validation and for their ability to get approval to market that pulse oximeter in most countries in the world.
All right, thanks, Jerry. So we're going to jump into the second poll question, and we'll throw that up on the screen. An SpO2 tester is a true simulation of a human finger. True or false? We'll let Lindsay get that up on the screen. So is an SpO2 tester a true simulation of a human finger?
Give it about 30 minutes and then we'll go through the responses.
Well, maybe not 30 minutes.
I was
30
just going to say, did I say
seconds.
minutes? I meant seconds.
I was
I
going to
don't.
let it slide.
Exactly. That's how you know we're real people doing this.
Yeah.
Still got a few more people voting here, so we'll let them roll in just a little bit longer. Okay, looks like we are rolling out most of our, wow, we have pretty high, I think about 75 to 80 people, 80% of the people have voted. So it's great engagement. Thanks you guys for participating with us here. So we've closed our poll and the responses say 60% say it's true, 40% say it's false. So we've got a good separation here. Let's talk a little more about that.
Jerry, take it
Well,
away.
according to the US FDA, Food and Drug Administration, who regulates even us as a manufacturer of test instruments, there is no true simulation of the human physiology when it comes to pulse oximetry. Well, why do they say that? Because they say in order to simulate it, one would have to produce a actual equivalent, a technical equivalent of the human physiology, not just the electrical signal and the optical signal. That means you would have to have some chemistry going on in a we'll say tissue equivalent, we mean blood equivalent, fluid or something, in order for it to be a true simulation. Well, that's not what we're producing. That's not what anybody is producing. We're producing something that is mimicking the light equivalent, the electrical and optical equivalent of what happens in the blood. So it is not a true simulation. It is rather instead a functional test of the oximeter. Okay, so the proper answer for the previous poll question is no, it is not a true simulation. Does that mean it's not valuable or effective in determining performance, the proper performance of a pulse oximeter, whether it's built into a patient monitor or standalone? No, this is perfectly fine to have a functional test. But remember that it's all got to be based on the R curve. And so you have to have the proper R curve for each brand and model of pulse oximeter that you're going to test. It just one size does not fit all.
Yeah, and it might be good to check out the white paper that we made. It's called "There's No Such Thing as an SpO2 Simulator" on the website. And that will walk through kind of the concepts that Jerry's talking about on this slide in more detail, and how that pertains to our ProSim Spot or Spotlight. Basically, those are our two SpO2 testers that are here on the screen.
So just to make it really clear to everybody, the Spot is the artificial finger that attaches to the ProSim 8. It is an optional accessory. And the ProSim Spotlight is a smaller, with fewer R curves built into it, version of the same thing. They're both functional testers all together.
I did also add that application note that you just mentioned to our handout section of the available content for you guys today. So just wanted to let you know that it's available right now as well.
Very good. Thank you, Lindsay.
Thank you, Lindsay.
Okay, so let's just take another look at this, and specifically talking about the R curve and the functional test. The functional testers, especially those that we, Fluke Biomedical, make and provide for you, pretend to be the finger. You see that on the left, you show us how the light would normally flow through the human finger. Functional testers, even though they are not simulating a finger, they can be fooled they can fool the pulse oximeter monitor into thinking its monitor is actually a finger. That's kind of what we're doing. They take the light input from the pulse oximeter, run it against the R curve that the user selects, and emit the right amount of light back to the detector of the pulse oximeter, and the result is shown on the pulse oximeter display. So what the oxygen saturation is that we simulate is what should be displayed on the pulse oximeter part of the patient monitor. The tester has 2 light detectors, as we said before, with a filter over one of them, so it can discern or identify the type of light and either red or infrared. As the monitor shines light into the top of the finger of the tester, red and infrared light is in succession delivered by the tester. It measures the amount of light, decides which light it is, red or infrared, calculates the test settings, from the test settings, how much of that light should be shining all the way through the finger, and then it sends that into the detector of the pulse oximeter, so it looks like and produces the right result on the pulse oximeter display. And that's how the technology works for functional test of pulse oximeters. This LED is wide enough a bandwidth of light to be detected by the monitor, and there only needs to be one light detector in the monitor that can detect either wavelength because the monitor knows which wavelength it's outputting, and therefore that is the wavelength it will be detecting at the time, the pulse oximeter. The tester is running heartbeat pulses that approximate the shape of the arterial pulse, because remember we said the oximeter must have pulsatile arterial blood flow. It has to have it, otherwise it can't give you a valid reading. So it varies the light according to the current amplitude of the pulse. So between pulses, it just gives DC light, and during the pulse, it gives the AC plus DC light, meaning it gives pulsatile information so that the pulse oximeter can respond properly. That's how the technology works for your tester.
Yeah, it's truly amazing how much thinking the functional tester needs to do in the background to be able to simulate that human finger as closely as possible.
Right. Okay, we can go on to the next slide, Mike. So why not your finger? So here's the thing. Even in some of the test procedures from the service manual of the pulse oximeter that you get from the manufacturer of that pulse oximeter, they will say in that test procedure, put the pulse oximeter sensor on your finger. Well, even though it's just a functional test, how do you know what oxygen saturation you are at personally? How do you know that? Do you? The answer is no, we don't know. That's the whole thing here. We don't know, and we need to have some credible, repeatable way to perform this functional test. So your finger's not good enough. The test has to cover the physiological range of variables for oxygen saturation from 30%, because below 30% O2 saturation, the patient is dead. Therefore, it doesn't matter whether you can measure below 30%. It doesn't. Up to 100%, and which we know would be a very healthy patient. The heart rate needs to be variable between 30 and 300 beats per minute because it has to be pulsatile. Finger thickness, skin color, light artifact, respiration artifact have to be able to be varied so that we can properly assess whether this pulse oximeter can still provide the right information, even when things make it difficult for that measurement to be made. And we also need to be able to vary perfusion and light transmission, that is transmissivity. So some of those things would be hard for a beginner to understand. So transmissivity, we, Fluke Biomedical, have characterized by more by the thickness of the finger than anything else. So that's how we can vary that. Well, what is the international standard? Because there is one for pulse oximetry. I've told you that the R curve is how we assess accuracy, but there is a minimum performance and safety standard under ISO, the International Standards Organization. The ISO 80601-2-61 is the minimum performance and safety standard for pulse oximeters. And it is what the manufacturers of pulse oximeters should be using in their design validation testing. It informs that along with their particular design and R curve into their service manual procedure for you to do the testing against. And it also informs us as we create test instruments that are going to properly be able to be used for getting a correct functional test. So we give you that through both the ProSim 8 with Spot accessory as well as in the ProSim Spotlight. So transmissivity is the percentage of light that is transmitted through the finger, selected by preset finger types in our test instruments that we provide you. Settings include dark, thick finger, medium finger, light, thin finger, and neonatal foot. Dark meaning equivalent of skin pigmentation, but described in a very, in a term, in a word that almost everybody could understand. Dark skin, thick finger. Medium finger, light skin, thin finger, neonatal foot. So these are kind of the things that could get in the way. There's one thing on here that we didn't talk about, and that is purple nail polish, really dark purple nail polish. That also prevents the light getting through the finger. So guess what? Clinically, they need to remove that nail polish in order, or pick a different measurement site for making the pulse oximetry measurement clinically. So those are the things that could get in the way. We don't simulate purple fingernail polish, but we are providing a way to simulate the other things to make sure the pulse oximeter can still do its job properly with the right reading, the right measurement, even as we pick these different settings. Okay. All right, pulse amplitude and perfusion. The range of pulse amplitude is 0 to 20%. What does that mean? That means think about it as the strength of the pulse. If you were to feel the pulse of a patient with your fingers on the blood vessel, you would feel the pulse, and the pulse would feel relatively stronger or relatively weaker. And so think about this pulse amplitude as how strong that pulse feels. So as a percentage, expressed as a percentage from weak to really strong, that is the range of pulse amplitude or perfusion. It varies the perfusion amount delivered, the amount delivered of the arterial blood in each heartbeat of the finger. All right, so that's kind of what those terms mean clinically, and we also use that in designing the test technology, the functional tester technology. The ratio of AC to DC components of light transmitted also affects how the pulse oximeter understands this pulse amplitude or estimate of perfusion. Okay, and the strongness or weakness of the signal clinically is actually the cardiac output, the strength of the contraction and the volume of blood that's being pushed out of the heart into to deliver the oxygenated blood to all of the cells in the periphery, in heads, toes, fingers, and ears.
Yeah, it's important to note how intertwined all of these vital sign parameters are within the human body. We've touched on quite a few in this, and we're trying to focus just on pulse oximetry. So it really is a complex system.
Right. Your body isn't just one organ. It's the combination of all of them. Let's talk a little bit about ambient light artifact. In the beginning with Dr. Severinghouse, one of the things that was troublesome in the first pulse oximeters that were provided to the marketplace that you could actually buy and use was artifact from fluorescent light fixtures, because fluorescent light flickers. I don't know if you've ever seen that, but it actually flickers by the frequency of the voltage of the mains power that's making that light happen. And that flicker, when you have, like here in the United States especially, a 60 hertz or 60 cycles per second frequency of the mains power, is similar to a 60 beats per minute heart rate. So what was happening is the light from the fluorescent lights above the patient in the OR or in the ICU, which is typically where we made these measurements initially, was causing wrong measurements to be made because it was responding to that light flicker and not to what was really happening in the absorption in the physiology. So sunlight is another thing that was happening that was really making it difficult for getting the right measurement. So one of the things that even today anesthesiologists will do is, if they're measuring on the finger, they have the finger clip or the single patient use disposable sensor, pulse oximeter sensor on the finger, they will take the package that came out of and put it over the top of that sensor to do what? To block out sunlight, block out fluorescent light, anything that has this flicker or would provide an inappropriate or artifact signal that would confuse the pulse oximeter in order to clinically get the right reading. Bilirubin is a little bit less of a problem, but it can be in infants if they're suffering from bilirubinemia, which is actually a liver problem, not a respiratory problem. And amplitude from 0 to 5 times the transmitted light. These are all artifacts that we want to make sure that the pulse oximeter, we need to understand how it would affect the pulse oximeter. So in the test instruments that we provide you, we give you ways to inject or produce some of these artifact signals so that we can see how can the pulse oximeter do in terms of its proper display and measurement and display of O2 saturation values. So these are some of the things that we can introduce in terms of light artifact and things that actually do get in the way in real clinical use.
All right, so why don't we jump onto the last artifact we have?
Respiration artifact. So as the patient breathes in and out, there's motion. And because the chest typically is rising and falling, and this has to do with the health of the lungs and the proper function of the lungs. So respiration artifact also can cause some problems in a pulse oximeter being able to actually make the proper measurement. By the way, it also gets in the way of invasive blood pressure measurement. It gets in the way of ECG measurement sometimes by introducing artifact onto the QRS complex. So the patient's breathing does affect the pulse amplitude, and so we provide an artifact setting for this from 0 to 5% of potential decrease in the pulse amplitude. That is just an artifact of how the patient's breathing is happening. And again, this is to really completely determine what is happening with the pulse oximeter. Now, when would we use artifact settings? Mostly, they would be used if we have a problem reported by the doctor or the nurse that says, hey, I'm just not getting really good readings on this last patient. Boy, we would like to know a little bit more about that last patient. But so what we do is we would then use the artifact, introduce the artifact to see what kinds of things were going on with this pulse oximeter for the clinical situation in which it was being used, and the doctor or the nurse complained it wasn't giving the right measurement. It's a troubleshooting tool.
Sorry to interrupt, Jerry and Mike. We are running out of time just a little bit here, so just wanted to give you a quick reminder of the time.
Okay, thank you. All right, R curves, I think we've gone through this a lot. R curves that are built into our oximeter readings are on the screen. The one that is a good place to start anyway with creating custom R curves is the Nellcore R curve. But all of these R curves are different. They're developed by the manufacturer. We've already talked about that. And you may find that you need an R curve that is not automatically built into the instrument, and we provide a utility that you can use to create an R curve that is useful and install it into the ProSim 8 as a custom R curve that can be used by you during testing of manufacturer brands and models of pulse oximeter that are not in the list. And that only applies to ProSim 8, not to ProSim Spotlight. The pulsed co-oximeter that we were talking about before, the best example and the first example of this was produced by a company called Masimo, and it is their rainbow oximeter. It uses 8 wavelengths of light to measure more parameters. The dyshemoglobins that are unable to carry oxygen, carboxyhemoglobin, which is typically filled with carbon monoxide, as we said, that gives pulse oximeters alone trouble, and meth hemoglobin, which is the oxygen, where the oxygen cannot bind to iron molecules in the blood. So these are just some of the samples. We have partnered with Masimo to provide a way to do proper testing of Masimo Rainbow pulsed co-oximetry, but you have to use a test sensor that you get from us. You cannot make the Masimo Rainbow assessment with the clinical sensor. That is the sensor that the doctors or nurses would use.
All right, so 2 more slides, Jerry. We'll just go through trends and then calibration. We'll skip the poll question.
That's fine. So trends. Reflective light technologies have been around for a while, but no one right now is doing a good job of functional test on reflective technologies. So that is something that a gap that needs to be filled. Personal monitors, like these are things that are used for you when you do exercise. Sometimes they're really designed for home use or personal use, but sometimes they'll creep into hospital or clinic use. They're really not medical devices. They're not designed to be medical devices. They're designed to be just estimates that you can use for your fitness training and things like that. Finger clips with integrated displays, accessories, and so forth for cell phones, smartwatches, things like that. These are not medical devices. Do not expect them to be able to functionally test to the requirements of the international standards, which apply only to medical devices. Okay, and the usage of light for more analysis. All right, and finally, calibration. we're going to come back to this. No, there is no primary calibration reference for pulse oximetry. They are all have too many variables to be able to provide a stable, reliable way to do specific calibration. Calibration means we are making assessments against a known standard, either a transfer standard from a primary standard or so forth. This is part of metrology. So the functional tester is not a simulator, and so the calibration is just making sure that our R curve is working right. Annual validation is still recommended. It's part of when you send your ProSim 8 or ProSim Spotlight back to us. It's going to be part of what we look at when, and it's called calibration, but it's not really so much a calibration. There might be some adjustments to displays and so forth, but that's about it. So it's all based on the R curve. All right. Pulse oximetry is a very important life-saving technology. It's as is pulse co-oximetry. It's a way to measure vital signs in a non-invasive way, because the less invasive we are, the sooner the patient gets well and can go home. It's destined to become even more common. It's in personal devices. It really already is there, in certainly in the United States and Western Europe. It's crucial during this current pandemic, the COVID pandemic, because patient oxygen saturation can be a key leading indicator, not only of the infection, but of the treatment plan to get that patient back to health. And so what you're doing when you're making functional testing of pulse oximeters is really, really important in order for the technology, the medical device, to be used in diagnosis and in proper treatment of patients so they can get well and go home. So just remember that. You are playing a key role in doing proper functional testing and understanding what is pulse oximetry. Okay, and finally, before we go to Q&A, you can learn a lot more and take a complete course about patient monitor testing, including pulse oximetry, in our Advantage Training Center, which you register for, and we keep track and we issue certificates of completion around. They are academic style courses, but not only there, but in our recorded webinars, others of our recorded webinars, and also in our video library. So there are all of these things, and they're all, just like today, free. You don't pay anything for these, because we provide all of this extra things for you to make sure you succeed and are able to do your testing effectively.
Thanks everybody for joining.
Thank you.